Okay, so today I'm going to be illustrating and explaining the different types of point mutations. And point mutation is when one base in a strand is replaced with a different base, resulting in various different effects throughout translation depending on which bases are changed. And in order to illustrate this, I'm going to use an example of a DNA coding strand, which is the top line, and then translated that into RNA strand, which is the second line, and this RNA codes for methionine, tryptophan, asparagine, alanine, and cysteine, and then the stop codon. But for the sake of time, I'm just going to use the one-letter abbreviations from here on. And then the third line is the first mutation that I'll go through. And all the way up until where there should be a stop codon, it looks the same. But where there's a stop codon, this adenine was transitioned into a guanine. And this type of mutation is called a read-through because instead of a stop codon, wherever the stop codon should be, there's an amino acid. So the strand continues translating until it finds a stop codon. Okay, so the next mutation example is another example of a transition, except this time from a pyrimidine to a pyrimidine. And it can be seen here at the third amino acid where cytosine is replaced with uracil. And this strand looks actually the exact same as the original non-mutated strand, and that's because it's a silent mutation, which means the bases change, but the amino acids that are coded for it don't actually change. So our mutation strand looks the exact same in terms of amino acids as the original mRNA. At first glance, this example already looks shorter, and it's because there's a transversion from cytosine to adenine. And this resulted in a stop codon. And the type of mutation is called a missense mutation, and it's when the base change results in an early stop codon. So instead of going all the way until a stop codon here, the mutation resulted in the early stop codon, stopping the strand short. So the next example mutation is actually the same length, 5 amino acids, as the original mRNA, except that there is a difference in the amino acids right here, where there's a C. And in this particular example, there was a transversion from guanine to cytosine. And the type of mutation is called a missense mutation, and it's when the change in a base, in this case G to C, results in a different amino acid being coded for. And here it's C instead of W.